Welcome to Night Shift Knowledge, a quiet place in the late hours where you can finally let go of the day. Take a moment to settle in, soften your shoulders, and allow the busy world to fade completely into the background. Tonight, we are going on a gentle journey through the fascinating corners of science, biology, and history. Before we begin, please know that the screen will fade to dark very soon. You can safely rest your eyes and leave this video playing as you drift off to sleep. I would also love to hear from you. Please leave a comment below with a general topic you would like me to cover in a future video. It is always a pleasure to read your ideas and shape this channel around what brings you peace. If you find comfort in this space, a like and a subscribe would be deeply appreciated. To ensure the next video quietly finds its way to you, please tap the notification bell. So as your breathing slows and your room grows a little darker, let your curiosity drift along with my voice. Let us begin. The giant viruses waking up. A solitary amoeba drifts through its watery world, a microscopic hunter in a vast, unseen ocean. It is a survivor, but today the hunter becomes the prey. A colossal entity, nearly the size of the amoeba itself, locks onto its victim. There is no escape. A portal opens on the invader's surface, and it begins to inject its genetic destiny, hijacking the cell's machinery from the inside out. This is not a scene from a science fiction thriller. It is not a computer simulation of the future. It is footage from a laboratory in France, capturing the moment something impossible happened. We have successfully brought life back from the dead. This invader, a biological machine built for one purpose, to replicate, had been locked in a deep, frozen sleep beneath the Siberian tundra. It sat dormant, entombed in silence, while human civilization rose and fell. For 48,500 years it waited. That means this virus last infected a living cell when woolly mammoths roamed the earth and Neanderthals carved their lives into stone, and now it is hungry again. The implications of this resurrection are profound. We are no longer just observers of ancient history, we are its active participants. And the key to unlocking this prehistoric door lies in a specific technique developed by a team of French scientists led by Jean-Michel Claverie. Claverie and his team didn't go looking for dinosaurs. They went looking for the soil itself. They traveled to the remote reaches of northeastern Siberia, a place where the ground never truly thaws. Here, the permafrost acts as a perfect natural freezer. It is stable, cold, and dark. Organic matter doesn't decompose here, it pauses. Mammoth bones, ancient vegetation, and yes, even pathogens are suspended in a state of suspended animation, waiting for the climate to change. But bringing a sample back is only half the battle. The real danger and the real discovery happens when you try to wake it up. You can't just drop ancient dirt into a petri dish. You need bait. This is where the science gets clever. The team uses amoebas as living traps. Amoebas are the universal prey of the microbial world. If a virus is capable of infecting anything, it can likely infect an amoeba. They call it the amoeba bait technique. By mixing the melted ancient permafrost water with a colony of amoebas, the scientists create a buffet. If a virus is present, it will wake up, realize it is surrounded by food, and attack. They watched. They waited. And then the amoebas began to die. The water turned cloudy with the ghosts of dead cells. Claverie knew they had caught something. But when they put the samples under the microscope, what they saw defied everything they thought they knew about viruses. Up until recently, we defined viruses by their smallness. They were the minimalists of nature, little more than a strand of DNA in a protein jacket. The influenza virus is tiny. You could fit thousands of them inside a single bacterium. But this is a giant. It is so large that for years scientists mistook it for a bacterium when they saw it under a low-power microscope. It is visible under a standard light microscope, something previously thought impossible for a virus. They named it Pandora virus, and the name fits. Opening it released a Pandora's box of scientific questions. These giant viruses possess genomes that are incredibly complex, carrying genes that do things we didn't think viruses were capable of doing. Where a flu virus might have 10 genes, Pandora virus has over 2,000. It is a genetic library, a biological archive from a time long forgotten, and most terrifyingly, it is fully functional. The experiment proved that these organisms do not just preserve their physical structure, they preserve their function. After nearly 50 millennia in the ice, the moment they encountered a warm host, they picked up exactly where they left off. They were infectious. This confirms a fear that climatologists and epidemiologists have shared for years. The permafrost is melting. It is thawing at an alarming rate, exposing layers of Earth that have been sealed since the Pleistocene epoch. If we can find a viable, infectious virus from 48,000 years ago in a routine sample, what else is down there? We have proven that the theoretical boundary for viral survival in ice is at least 500,000 years. But is there a limit? Is there a point in deep time where the genetic machinery finally degrades beyond repair? Or are there things buried deeper, older, and perhaps more dangerous than we can imagine? Some scientists claim they have found evidence of something much, much older. And if they are right, we are just beginning to understand what is waking up. The 250 million year debate. In our previous chapter, we witnessed a microscopic execution, a billion-year-old dance between hunter and hunted. We saw the virus, that ancient amphora-shaped entity, pierce the cell wall of an amoeba, hijacking the machinery of life to replicate itself. It was a reminder that even in the smallest realms, existence is a brutal game of survival. But that encounter took place in the present day. It raises a haunting question that echoes across deep time. How long can these biological entities actually survive? We know viruses and bacteria have been around for billions of years, but is there a limit to their endurance? Can life pause itself, waiting out the eons, only to wake up in a world that has long forgotten it? The answer to that question might lie buried beneath the deserts of New Mexico in a geological formation known as the Salado. Here, within the Permian Basin, lies a graveyard of an ancient ocean dried up and compressed under the weight of millions of years of history. In the late 1990s, microbiologist Russ Vreeland and his colleagues were drilling into this formation when they pulled up something extraordinary: salt crystals. But these weren't just ordinary rocks. Inside the crystal lattice, trapped like microscopic insects in amber, were tiny pockets of fluid inclusions. Vreeland believed these fluid pockets were pristine time capsules, sealed off from the outside world since the Permian period. That was 250 million years ago, before the dinosaurs, before the breakup of Pangaea. In the lab, the team performed a procedure that sounded like science fiction. They drilled into the crystals, extracted the ancient fluid, and placed it in a nutrient-rich medium. Then, they waited. To the shock of the scientific community, something woke up. Bacteria began to grow. Vreeland claimed he had revived Bacillus permians, a microbe that had been sleeping for 250 million years. It was, if true, the oldest living thing ever found. But in science, an extraordinary claim requires extraordinary evidence, and almost immediately the skepticism began to pour in. Critics called it the contamination war. The central question wasn't about the bacteria's ability to survive, but about the integrity of the vault. The argument against Vreeland was simple. Salt is not impermeable. Over 250 million years, geological shifts occur. Fluids move. Cracks form. Critics argued that modern bacteria from the surrounding groundwater could have penetrated the crystal, settling into those fluid inclusions just a few thousand or even a few hundred years ago. If even a single molecule of modern fluid entered the crystal, the 250 million-year-old label would be wrong. Proving a negative is notoriously difficult. How do you prove absolutely that no contaminant ever touched that sample for 250 million years? The burden of proof rested entirely on Vreeland's team, and the more they tried to defend their sterility protocols, the louder the chorus of doubt became. While the debate over the New Mexico salt raged, another team of researchers was looking for answers in a place far more stable, far colder, and perhaps far more reliable. They went to the bottom of the world. This is Beacon Valley, Antarctica. It is one of the driest and coldest places on Earth. Here, the ice doesn't melt. It sublimates, turning directly from solid ice into vapor, leaving behind a hyper-dense, ancient record. In 2007, scientists led by David Marchant drilled into the permafrost here. They weren't looking at salt crystals, they were looking at ice. And they found bacteria within the ice that was dated to be 8 million years old. The environment here is key. Unlike the porous salt deposits of New Mexico, the Antarctic ice is remarkably stable. The temperatures are perpetually below freezing, and the lack of liquid water means there is no medium for modern contaminants to flow through the ice and contaminate the samples. This is distinct from the famous Siberian permafrost, which is thawing rapidly. The Antarctic ice in Beacon Valley is like a deep freeze that hasn't been opened. It suggests that while the 250 million year claim from New Mexico remains hotly contested, the survival of life for millions of years is a verified fact. We now know that life can shut down its metabolism, repair its DNA damage sporadically, and wait. Whether the limit is 8 million, 50 million, or Vreeland's 250 million, the door is opening. We are learning that the past is not as dead as we thought. These organisms are not fossils. They are not stone impressions. They are potential. They are biological entities that have outlasted mountains, oceans, and civilizations, waiting for the moment the environment becomes hospitable again. And we are making it hospitable. Through climate change, through deep drilling, through the disruption of ancient permafrost and salt mines, we are the ones waking them up. The debate about the exact age of these microbes will continue in the halls of academia. But out here, in the real world, the timeline matters less than the result. We are unlocking doors that have been closed for eons. The question is no longer just, can they survive? The question is, what happens when a microscopic predator that hasn't seen a host in 250 million years suddenly meets us? Pandora's Box Protocol. We left the last chapter witnessing a microscopic execution, a billion-year-old dance between hunter and hunted. We saw the virus, that ancient silent stalker, pierce the cell wall of an amoeba, hijacking the machinery of life to replicate itself. It was a reminder that in the smallest realms, existence is a brutal game of survival. But what happens when the arena of this battle changes? What happens when the ice that has held these ancient combatants in check for millennia begins to melt? The stage is set, not in a petri dish, but here, in the vast, thawing expanse of the Arctic Circle. In the summer of 2016, the Arctic experienced a heat wave that defied historical norms. temperatures soared, melting the top layer of the Earth that had remained frozen since the dawn of human civilization. But the heat didn't just melt ice, it woke something up. The Yamal Peninsula, a remote stretch of northwestern Siberia, became the epicenter of a biological nightmare. Thousands of reindeer, nearly 250,000, dropped dead, their bodies scattered across the tundra like fallen leaves. But the terror didn't stop at the herds. The pathogens spilled over into the human population. Dozens of people were hospitalized, and a 12-year-old boy died. It was the first anthrax outbreak in the region in over 75 years. The question wasn't where it came from, the question was how it had survived. The culprit was Bacillus anthracis. While we focused our fears on viruses in the last chapter, here we were reminded that bacteria have a far more terrifying trick up their sleeve. When conditions get tough, bacteria don't just die, they hibernate. They transform into spores. Think of a spore not as a living organism but as a seed with an armored shell. They are biological bunkers. They shut down their metabolism and become impervious to cold, heat, drought, and time. They can wait for centuries. The 2016 outbreak traced back to a reindeer carcass that had died decades prior and frozen into the permafrost. The heatwave thawed the cemetery of ice, releasing the dormant spores back into the air and water. The anthrax didn't need to evolve, it just needed to wait for us to open the door. This was the smoking gun. It proved definitively that thawing permafrost is not just a geological curiosity, it is a biological time capsule. And while anthrax is a known enemy, familiar and treatable with antibiotics, it serves as a stark warning. The permafrost is leaking. But nature is not the only one breaking the seal. While global warming provides the slow, relentless creep of thaw, human industry is delivering the sledgehammer. We are not just waiting for the ice to melt, we are digging straight through it. The Arctic is rich in resources. Oil, gas, gold, and diamonds lie beneath the frozen earth. To get to them, we must excavate layers of soil that haven't seen the sun in over 120,000 years. We are effectively tunneling into the past, bringing ancient geological strata to the surface at an industrial speed. Natural thawing is a gradual process, giving ecosystems time to adapt, or at least to filter what is released. Mining, however, is violent and abrupt. It churns up deep, ancient sediments and aerosolizes them, creating dust clouds that can carry pathogens for miles. Jean-Michel Claverie, a virologist who has spent his career hunting these ancient pathogens, warns that we are treating the Arctic like a candy store. He argues that these industrial sites are the canaries in the coal mine, but not in the way we usually think. Usually, the canary warns us of danger before we die. In this case, the industrial sites are the ones breaking into the toxic chamber first. By drilling into the deepest layers of permafrost, we are the ones disturbing the ghosts, and we are bringing those disturbed ghosts right into our own backyards. And this brings us to the true heart of the fear, the unknown. Anthrax is scary, but it is a known quantity. The real terror lies in what we haven't catalogued yet. Claverie and his team have done the unthinkable. They have revived viruses that have been frozen for 30,000 years. They found Pandora viruses and pithoviruses, massive entities so large they were initially mistaken for bacteria. While these particular ancient giants only attack amoebas, their resurrection proves that viral DNA can remain infectious for eons. Consider the timeline. For hundreds of thousands of years, the Arctic was inhabited by various hominids, Neanderthals, Denisovans, extinct human relatives. When they died, they often died of disease. Their bodies, frozen in the permafrost, are potential tombs for ancient pathogens. Smallpox, the flu, unknown plagues that wiped out entire tribes. These pathogens could be lying dormant, perfectly preserved, waiting for a new host. And for the first time in millennia, a potential host is walking right above them, us. Our immune systems are built on a library of memories. We have defenses against diseases we have fought before or that our ancestors fought, but we have zero memory of a virus that died out during the Ice Age. To our modern T cells, an ancient pathogen would be completely alien. It would be an immunological naivety. We would be like the indigenous populations of the Americas encountering smallpox for the first time, devastated by a biological invader against which we had no defense. The scientific consensus is chilling. It is not a question of if a viable, dangerous pathogen will be released from the permafrost. It is simply a question of when. We are standing on the precipice of a new era in biology. We have mastered the ability to look back in time, to decode the genomes of the past, and even to revive them. But this power comes with a profound responsibility. The delicate balance between scientific discovery and existential risk has never been more fragile. We are racing to understand these organisms before they escape, even as our industrial activities and our warming planet accelerate the release. We are waking up a world that slept long before us. The permafrost is not just ice, it is a vault of biological history, and we are melting the key. But the threat doesn't end at the edge of the ice. If the permafrost is the pantry, the global water cycle is the delivery system. And that raises a terrifying question about what might be moving through the rivers and oceans right now, undetected. We have reached the end of tonight's topics, and it is time to let your mind completely unwind. Whatever you learned tonight, let it fade into a soft hum as you drift off. Thank you for being a part of Night Shift Knowledge and sharing this quiet space with me. Remember to leave a topic suggestion for next time if you are still awake, but for now, just rest. Sleep well, and I will be here the next time you need to quiet your mind. Good night.